We continue our lecture on amino acids and proteins, and in part 1, we finish objectives 1, 2, and 3. Part 2, we're going to cover objectives 4, 5, 6, and 7. And that is, we're going to deal with amino acid pKa's and ionization of amino acids, the nature of the peptide bond, protein folding, and the use of chaperones to help protein folding, and then protein structures. Amino acid pKa's. So the pK values for amino acids are usually given in a table such as this. Now what you need to realize is when you look at an amino acid like glycine and you have two pKa values, one is going to be low and the other is high. Remember, pKa is the pH at which the hydrogen ion comes on or off, begins to come off. All right. More specifically, the pKa is the pH at which the amount of base and the amount of acid form of an amino acid concerning a specific group are at 50%. Okay, and so any amino acid that has two pK values, that means those amino acids have two ionizable groups. The low one usually refers to carboxylic acid. High one usually refers to the amino or the base. An acid has low pKa, a base has high pKa. Then you come to some that have very high pKa values, the third pKa values, right? These are very unusual, so I'm going to skip. And the one that I want you to focus on are aspartic acid, glutamic acid, arginine, lysine, histidine, and so forth. Okay? So look at for aspartic acid. It has three pKa values. And we're specific in saying the alpha carboxyl is 2.0, alpha amino is 9.9, but the alpha group, and that is, it has to be carboxylic acid because the pKa is low, is 3.9. Look at glutamic acid. Again, two low ones refer to carboxylic acid, and one high one, which is the amino group. Okay? Look at lysine. One low one, which is carboxylic acid, that's the alpha, and two high one. The alpha amino is 9.2, and the R amino group is 10.8. Now, the only unusual amino acid that you can't help but commit to memory is histidine. Look at it. The pKa1 is usually a carboxylic acid, has a pKa of 1.8. pKa2, the amino group, is 9.2. But then there's an R group that has a pKa of 6.0. Now, initially, one would assume that since it's less than pH of 7, that it must be an acid. Well, this is unusual because it's not an acid. It's actually an amino group. So that's very important when we begin to look at amino acid ionization. Now, anytime that you need to work with ionization of amino acids, I will give you a table such as this to use. So you don't need to memorize this. Ionization of amino acids, let's cover that. The number of pK for an amino acid, as we stated, indicate the number of ionizable groups. Ionizable groups, when they're protonated, for the acid, it's neutral. When it's deprotonated, it becomes negative. For the base, when it's protonated, it has a positive charge on it. When it is deprotonated, it becomes neutral. So you cannot just commit this to memory and say it, when a functional group is protonated, then it is no charge. Deprotonated, it is negatively charged, because it depends. If it's an acid, that is true. If it's a base, that's not true. So kind of look at it, look at the ionization, and look at the ion that is formed. Ionizable groups are either on the alpha carbon for all the 20 amino acids, that is the alpha carboxyl and alpha amino group, or they are on the R group, that is true for 9 out of the 20 amino acids. So 9 of these amino acids have ionizable groups on the R group, whereas the rest of them do not. We can calculate ionization of amino acid based on their pKa. All right, let's look at the example that is given here for aspartic acid. Look at aspartic acid. The alpha amino has pKa of 9.9, alpha carboxyl has a pKa of 2.0, and the R group carboxyl has a pKa of 3.9. Okay? Now, at pH 1, I want you to look at something for me. At pH 1, you're below the pKa of the carboxylic acid. So, hydrogen ion is not coming off yet. Remember, this is what hydrogen ion comes off. So, this is COOH, no charge on here. At pH 1, you're way below the pKa of the amino group or the hydrogen ion, or the hydrogen will come off. So it is H3N or NH3 plus. So no charge on carboxylic acid, plus on the amino, R amino. Then we look at the carboxylic acid on the R group. Again, you have to go up to pH 3.9 in order for the hydrogen to come off to generate a negative charge group right here. So here hydrogen ion is on at pH 1, 0 charge, no charge. Here hydrogen ion is on at pH 1, no charge. Here hydrogen ion is on, +1. And so at pH 1, the net charge on that amino acid is +1. Now let's go to the other extreme, 14. That is, at pH 14, all the pKa's of all the ionizable groups have been exceeded, which means that hydrogen ion that needs to come off will come off since you have exceeded the pKa value. Hydrogen ion has come off right here to generate COO minus. When it's off of here, generate COO minus. When it's off of here, there's no charge because you drop the hydrogen and this becomes NH2, no charge. So at pH 14, the net charge on the amino acid is -2. Now let's go to pH 7.4 and make it more interesting. At pH 7.4, you have exceeded the pKa for the alpha carboxyl and the R carboxyl, which means they're generating the -1 here and -1 here to make -2. The amino group, on the other hand, has not been exceeded. The pKa of 9.9 has not been exceeded by 7.4. So hydrogen remains on here, it's positively charged. So +1, -1, these cancel themselves out, -1. So at pH 7.4, the net charge on that molecule or that amino acid is -1. Let's look at ionization of, let's look at ionization of aspartic acid using some other pH. So let's look at pH of 2.1. 2.1 has exceeded this, so it's negatively charged. 2.1 has not come up to that 3.9, so this is on, no charge. And pH 2.1 has not exceeded that, so positively charged. So you got positively charged, negatively charged, 0 charge for a net charge of 0. Okay? At pH 4.4, you have exceeded this, which is -1, exceeded this, which is plus -1 is -2. You have not exceeded this, plus 1. So plus 2 added to plus 1 is -1. At pH 4.4, the net charge on that molecule is -1. At pH 8.0, at pH 8.0, you've exceeded this, that's -1. You've exceeded this, that's -1. To add them together, that's -2. You have not exceeded this, so it's still plus 1. -2 plus 1, that is -1 at pH 8.0, right here. Okay? Now, let me point it out. A pH at which the net charge on the molecule or the amino acid is equal to 0 is called the isoelectric point. And that's a very precise, very specific pH. It's the pH value at which there's no net charge on that molecule. And right here, it appears that 2.1, and we can play with that, see what would happen at 3.1 and what have you. But it's close to 2.1, that's the isoelectric point for aspartic acid. Now, let's go on to the peptide bond. The peptide bond is a bond that is formed between the alpha carboxylic and the alpha amino group to remove water and to generate this peptide bond right here. You have this carboxylic acid that is part of this amino acid. You have this amino group that is part of the next amino acid. When you remove water, these two interact to form a peptide bond. A peptide bond is between two amino acids, between the alpha carboxylic and the alpha amino group, and it occurs through the loss of water or dehydration. This is what happens when you link the subunits of proteins together, or the components of individual component of proteins, which is amino acid, together. In order for them to link into a polymer of amino acid to make proteins, you must generate peptide bond between a chain and amino acid. Now, peptide bond is it's a single bond right here, but it doesn't move around freely, and the reason for that is it has double bond characteristic right here. It's not truly single bond, it's not truly double bond, but it's somewhere between a single bond and a double bond. When you form peptide bond, you need to realize that there is directionality. All right, let's look at these short peptide, these oligopeptides right here. One begins with methionine, followed by alanine, followed by another alanine, followed by glycine. Now, the dashes represent peptide bonds. Now, when you look at this, you have a left-hand side, which is for DNA is 5'. The right-hand side is 3' for DNA. Well, for protein, it's called the amino N and the carboxylic N right here. So, look at this. Methionine, alanine, alanine, glycine. Now, look at the other one. Glycine is at the amino N, followed by 2 alanines, followed by methionine at the carboxylic N. Now, according to amino acid components of these two oligopeptides right here, they are identical in their amino acid contents. However, these two oligopeptides are as different as night and day, because look at it. The amino terminus of the top one is methionine, whereas the amino terminus of the top one is glycine. The C terminus of the top is glycine. The C terminus of the bottom one is methionine. So if these were two functional tiny, tiny, tiny proteins, these oligopeptides, they would have completely different chemical properties, perhaps different functions, because they're not the same proteins. They have the same makeup, amino acid makeup, but they're completely different when it comes to directionality, the amino and the carboxylic acid. Okay? So this is what this point said. The two peptides have different amino acid sequence. Two peptides have different ends, but two peptides have the same amino acid composition. So sequence is different. Ends, yes, sequence different. The ends are different, but the compositions are identical. Therefore, they're different oligopeptide and probably have different function. Here is a cartoon of the peptide bond and its formation. And so, see that there's a loss of water, dehydration, and formation of the peptide bond. Okay? Now let's talk about protein folding. Protein folding. How do protein fold into their three-dimensional functional structures? Well, we know that when proteins are synthesized in vivo, they fold correctly. The newly synthesized proteins are not folded immediately after they're synthesized or as they're synthesized, but short period of time after that, they begin to fold into their three-dimensional structure. Now, how do they do that? And what causes that folding to occur and to occur without any problems? There was one hypothesis that was postulated or that was formed initially, and that is that when you synthesize protein in an aqueous environment, that protein is going to experiment with all the possible conformations until it reaches the conformation that has the lowest energy level or the lowest energy state. And that is, and I heard this when I was in graduate school, that a protein could actually experiment or sample thousands upon thousands upon thousands of different conformations before it arrives at the right conformation. And so here, and Vincent performed, and his colleagues at the NIH performed an experiment that is a hallmark experiment in protein folding, and that is that they took a tiny protein right here, and they begin to reduce the disulfide bonds right here to form free sulfhydryl groups and destroy the three-dimensional structure of this protein. And then they added 8 molar urea, and they basically denatured this protein. It goes from 100% activity to 0% activity right here. But then if they were being careful in, number one, removing the urea first, followed by oxidizing of these sulfhydryl groups to form the disulfide bridges, it goes back into the native state. You go from native to denaturation, 0 activity, and then when you renature, 0 activity to 90%, almost full activity. Then they did another experiment, and that is, if they were to, again, reduce it and add 8 molar urea, reduction is, breaks the disulfide bonds, using 8 molar urea destroys all the other interaction to make it a denatured and non-functional protein. But now look at the order of events that were performed. They oxidized the sulfhydryl groups instead of removing the urea to allow the protein to come together before oxidizing the sulfhydryl groups. And you see that when they did that, look at what happened. Yeah, the protein came together, all right, but the structure is completely different from the native structure. They call it scramble, and it only had 1 to 2% activity. In other words, it is important that you remove the urea first to bring the protein back together to a loose structure prior to oxidizing the sulfhydryl groups to form the disulfide bond. The idea is, if you don't do that, if you don't allow the protein to come together loosely so that the sulfhydryl, S can find their correct partners, you're going to end up with a completely different structure of protein. Here, we can say A and D, A, B, C, D, A and D bind together, C and B bound together, right, to form this. Well, if you don't bring the structure together loosely before you allow the sulfide bonds to be formed, it is possible, and this is what the experiment shows, that the SH groups or the oxidized S will actually pair up with the wrong one that it doesn't need to pair up with. For that reason, you have secondary or the structure of the protein that is different from the native structure, and therefore the activity is about 10 to 5% of the native activity. And this is just talking points, right? Reducing disulfide, denaturing with 8 molar urea, the order in which you reduce and you oxidize really made a big difference on whether or not you will get full activity or just a tiny amount of the activity back. All right. And the conclusion of Anfinsen and his colleague experiment is that, yeah, there's some spontaneous folding. Yes, spontaneous folding is involved, but the protein sequence, the sequence of amino acid, actually determines the functional structure of the protein. And even at that time, after this landmark experiment that we can look back and hindsight is 2020 and we can understand it, even after that, there was still a lot of controversy. Well, how do proteins fold? Well, do you remember? We stated the thermodynamic hypothesis, and that is that the protein experiment with all the possibilities that they must be tried until it adopts the most stable or the lowest energy one. Well, that was the first possibility. The other possibility is this, that perhaps based on Anfinsen's experiment, perhaps the protein does not sample all the hundreds of thousands of different conformations that are possible until it comes to the most stable conformation. Perhaps the protein goes through just a number of intermediates that are loosely related to the final conformation. And so it goes through 1, 2, or 3 intermediates, and then it comes to the final conformation. Really, this was not clear until Leventhal proposed the Leventhal paradox. And that is, if the thermodynamic hypothesis is true, then we can calculate the possibilities this way. Let's assume that a tiny protein that has 100 amino acid residue is under consideration. And let's assume that each amino acid will sample only 3 different conformations. Only 3, not the endless possibilities that was proposed under the thermodynamic hypothesis. Only 3, each of the 100 amino acids will sample 3 possible conformations. The number of structures that are possible is, take a look at it, 3 times 10 to the 100. Wow. That is something, right? Okay, so 3 to the 100 power, or number of structure is 5 times 10 to the 47th power. Now, let's assume, for the sake of argument, that it takes 1 times 10 to the minus 13 second. That's a very fast event right there. From structure to structure. Now, search time will be the number of structures that are possible, 5 times 10 to the 34, multiplied by the time that it takes each one of that conformation to be experimented with, which is 1 times 10 to the minus 13 second. If we were to convert that to a year, it would take 1.5 times 10 to the 27th years. Now, how many of us would be willing to wait on a protein that is being synthesized in our bodies because we need it and we need it immediately? How many of us will be willing to wait 1.5 times 10 to the 27 years? Impossible, right? So Levinthal's paradox began to open the eyes of scientists everywhere and said, that is correct. It's not endless possibilities. It must be just a few, 1, 2, or 3 conformations that are experimented with before the protein arrives at the most stable structure. I'm going to go past this. And so, how does protein fold then? Well, protein folding has an assistance to help it. It has a number of enzymes. We call this folding enzymes that consist of protein disulfide isomerase. You know what this does, right? Protein disulfide isomerase is an enzyme that enables the disulfide bonds to be formed between the right partners within each protein. Wrong partners will lead to the formation of a completely different structure of the protein. Another enzyme is peptidyl prolyl isomerase. And this is an isomerase that makes sure that the right conformation, if it's cis, it has to be cis. If it's trans, then it will make it trans, the conformation of the proline residues in the protein structure. This isomerase makes sure that whatever it calls for, whether it be trans, which is most of the proline residues are in a protein, or a few of them which have cis, that it is the right conformation, either cis or trans. And then you have other chaperone proteins, a number of chaperone proteins. And when we talk about chaperone proteins, we're talking about proteins that, when the protein is synthesized, chaperone proteins begin to surround that protein molecule and provide an environment for that protein molecule to fold and keep it from other protein molecules that it might interact illegitimately with. One of the interactions that occurs between protein molecules is hydrophobic interaction. If they interact, they will fall out of solution, precipitate in the cytosol, and that's not good. So to prevent illegitimate interaction between newly synthesized proteins, chaperones serve the role of protecting those proteins and providing an environment for them to fold unto completion, where there's no more chance for them to interact with other protein and fall out of solution. So protein folding then is like this. You have the unfolded primary structure, right? It's unfolded right here. And then you have an intermediate called molten globule, which is very similar. It's loosely similar, almost identical to the final native structure, but it's looser than the native structure. It lacks a compact three-dimensional structure of the stable, the native structure. So you have unfolded protein that is newly synthesized. It begins to fold with the help of enzymes and chaperone proteins into a loose replica of the native state, and then it eventually becomes that compact structure of the native state, which is fully active right here. And that's what protein folding is. One of the telling signs is that a few years ago there were about 39,323 proteins, different proteins that were deposited on this protein data bank right here. If you get a chance, please go to this website. And yet, among these nearly 40,000 proteins, there were only 1,000 unique folds among those proteins, which means that they share a lot of intermediate states right here among them. Okay, very similar structures. So over 90% of the structures deposited have similar folds to the others. And so protein folding is divided up into 3 steps. Step number 1, within a few milliseconds, you get formation of the molten globule. Step 2, which lasts up to 1 second, native elements and tertiary structures begin to develop. Step 3, single native structure is formed. And this is the one that has the least or the lowest energy level. It's very compact, where the hydrophobic groups are sequestered to the interior of the proteins, and the hydrophilic groups are exposed to the outside. And this is the unfolded. This is the depiction of the stages. You have unfolded primary sequence of the protein, fast to formation of loose molten globule, which is similar to the native structure, but there's a slower stage where it becomes that compact native structure right here. And this is the structure at which the protein is very functional, carries out its function. Again, if you were to trace the energy, unfolded has this energy, molten globule has lower energy. It kicks it up to a transition state. Remember, this is very fast. It goes to a lower energy level, kicks it up to the high energy state right here, and then decays down to the lowest energy level where the protein begins to fold is to its three-dimensional structure. One of the examples is RNase, okay, bacterial RNase, and that is had 110 amino acids. It has a number of helices right here, a number of loops right here, and we're going to study what these structures are. And it has a number of beta sheets right here. All make up the three-dimensional structure. And experiments, the reason why this is good is that when they work with RNase, the bacterial RNase is the one that chews up RNA. That's why we tell you in the lab to be very, very careful in handling all the reagents that are going to be used for RNA isolation, because you cannot inactivate RNase very easily. And that's why they use RNase in these studies, because it's a very hardy enzyme. The folding process goes from unfolded state to one single intermediate, the molten globule, and then it goes to the native state right here. So this is an example that instead of experimenting with thousands and thousands of different structure, it only goes from the unfolded to an intermediate to the lowest energy level, which is the most compact. I'm going to go ahead and stop right here, and we'll come back and finish up the lecture beginning with protein structure.